everybody. Welcome to today's lecture, where we will be focusing on DNA structure and replication. So let's go ahead and get started. So give me 1 second. Let me set up my screen. So there are some of our learning goals for today's lecture, which includes looking at the structure of DNA, the scientists that were involved in the process, including Watson and Crick, that created the three-dimensional form of DNA. Look at the main structures and components of DNA, which are basically nucleotides, the format of the double-stranded DNA and how that is created. And then we're going to dive into concepts like DNA replication, looking at the steps involved in the process, and then looking at the process called PCR, or polymerase chain reaction. And then what is that we can basically use to in our daily lives and in the healthcare field that could benefit us by using DNA and genetic technology. So the first thing that I want you guys to understand is we have done a number of experiments over the past century, I want to say, the past 70 to 80 years that basically help us step-by-step understand the genetic material within our body better and better. So one of the first experiments was done by Francis Crick. Well, I'm sorry, but not one of the first experiments, but one of the experiments that was done by Francis Crick, and he actually, from those experiments, he made the following comment. He says, a genetic material must carry out two jobs. One job is to duplicate itself, which is the process of replication, which we will be focusing on for today's lecture, and then also control the development of the rest of the cells in the specific way. And when we talk about this, what our focus is basically gene expression, which is, again, if you kind of recall from the previous, is basically encoding for proteins. And DNA, which is our molecule for today, fulfill both of these requirements, replication and gene expression. Now, one of the first people that noted information about the DNA is Meissner in 1871. He was actually a physician as well as a biochemist, which absolutely fascinating. But he was able to isolate nuclei, which is basically the nucleus of multiple cells, that's nuclei, of the white blood cells, the WBCs, cells responsible for your immunity, by basically collecting pus, which is what's released during the infection, and assessing that. He found these acidic substance, hence nucleic acid, that has also contents of nitrogen and phosphate. And we'll look at these and why this information was so important for us to understand the structure of DNA. So basically, it has acidic component, it has nitrogen, and it has phosphorus. He called it nuclein, so that's the first name he came up with. And then as time passes by, he changed the name to nucleic acid, which is what we are currently calling. And again, nucleic acid includes things like DNA or RNA. And it has other things like ATP, also considered a nucleotide, more specific, not nucleic acid. We have Archibald Garrett in 1902, so we're talking about over a century ago. He was also a physician in this case, and he was able to connect and link inheritance in the inborn errors of metabolism with the lack of particular enzyme. So basically, he said, the reason you don't have a particular enzyme, which means there is lack of function, right? Lack of function, because enzymes are responsible for chemical reactions. The reason for that, and the reason we cannot complete the metabolism process, is because there is a genetic component to it. So again, we're connecting DNA to proteins. That's the connection he was trying to make in this case. Griffith, the concept of Griffith, he focused on the concept of transformation. So again, we're looking at over almost a century ago, Frederick Griffith, who was a microbiologist, so we're kind of getting into the field now, work on Streptococcus pneumonia, which is basically the bacteria that gives you pneumonia. And it's found that there's two types, first of all. So there's two types of this bacteria, which is really important for our understanding of his experiment. The type that's referred to as S and the type that's referred to as R. So a smooth, S for smooth, it enclosed inside a polysaccharide capsule. So if this is like your bacteria, you have a coating around that, which is the capsule. And if it's the rough version or type R, there is no capsule. So you only have the cell, no capsule around it. Now, this is important basically of how it gets transformed from one organism to the next organism. And here's basically what we're trying to explain. So this is very important, guys. And again, we're kind of going back to that whole idea of control versus experimental when we're designing an experiment and following the scientific method. So if you guys worked on your Canvas Post 1, this should sound really familiar since you have explained those concepts and within your example came up with your own control and experimental. So what did he come up with? He first came up with a control experiment. So the first control was that he had the type R, which doesn't have the capsule, being injected to a healthy mouse. So your mouse is healthy. And nothing happens to the mouse. The mouse was perfectly happy. It survived. So this control was basically, you saw that the non-virulent version of this bacteria does not cause harm to the mouse. Now, the second control he did is he actually used a smooth version or type S, which was virulent. Virulent basically means that it's going to cause infection. And he noticed that the mouse dies. So you already have two very strong experiments where non-capsulated version of this bacteria has no effect on the organism. And then the virulent, the one with the capsule, will cause the death of the mice. Now, in his experiment, he first took a smooth type, which was the virulent, and then heat killed it, which basically means expose these cells to significant amount of it. I'm sorry about the sound. Expose it to significant amount of heat and basically destroy these cells. And as it injected to the mouse stayed healthy, which means we basically got rid of these cells. The next thing that it did, it took the rough virulent, which is the type R, which is normally wouldn't kill it, and added the smooth virulent type, which is the S. So now you're combining both of these. So now it means that there is a allowing for transformation of something that was not harmful to something that can cause death. So what he noticed basically is when he combined these two cells, the type R and type S, which has been heat killed, that still caused the mouse to die. When he pulled the cells out, he noticed that the cells are showing as type S, as the virulent type, right? So virulent type, it means that by combining these two together in part D, you actually created something that now can cause harm to your organism. So that means type S bacteria transferred its killing trait to the type R bacteria, which was non-killing. And this is really important, which again implies that the molecules that basically cause this virus to be harmful has been transformed to has been basically moved from the non-virulent to the virulent one and causes lethality on that type of bacteria. So you're transforming a non-killing bacteria, which is type R, to a killer bacteria, which is type S, by combining them together. So that genetic material has been transferred between the two types of cells. Now, there was another research which was done in 1940s by a group of researchers, and you have the name MacLeod, McCarty, and Avery. And these are also physicians. So again, they're directly working with and looking at data. And they treated a lice version of a S bacteria with two things, protease and DNase. Now, what are protease and DNase? And here is the definition. If you see the word ase at the end, first of all, it means we're talking about an enzyme. So protease implies it's an enzyme with the goal of destroying proteins made up of peptide chains. So all it does is literally chopping up your proteins to little pieces and therefore taking its functionality. DNA also, if you kind of think about it, use the same logic. DNA ACE implies that it's going to destroy or break down the DNA. So it destroys the backbone of DNA and leading to degradation of your DNA, which is again not a good idea, right? It's not a good thing to happen for yourself. So I'm going to just change my color. So this is a very important information, guys. When they only treated it with DNase, it prevented the transformation, which means when I destroyed the DNA, when I removed the DNA, there was no transformation of that medical condition, right? But if I was treated with protease, nothing changed. Everything was still okay. Okay, so there was a difference in terms of DNA was the component that allows for the characteristic to be transformed. So again, we're looking at transforming principle of genetic information. Protein didn't have that capacity, but DNA did. So again, it can convert something that's rough to a smooth. So from non-virulent, I'm going to keep saying that, to the virulent. So here is a visualization of the experiment. You break your cells open, you treat it with protease. So only DNA is going to be present. Because the DNA is present, you will kill the mouse because the DNA will be affecting the cell, and the cell still have the ability to maintain their genetic information. And this one, when you have protein only present, but remove all of the DNA, then these cells do not have that transforming power going from non-virulent to virulent, and therefore mouse would live. So proteins do not have transforming power. DNA has transforming power. The other research that was done is that protein is not the hereditary components of the molecule or of the cell. So again, at this point, when we're looking at these experiments in 1950s, you have an understanding that the cell has proteins. And you have an understanding that cells have DNA. But you're still trying to figure out what actually gets passed on from the first generation to the children. So for this research, there are microbiologists that carry out this research. First of all, they dealt with E. coli. So E. coli is a very common bacteria. You probably have heard of them in terms of infections or food poisoning, but it is a very common bacteria, and it does cause infection. So they use E. coli bacteria that was infected by a virus. So if you don't know, viruses are much smaller than the bacteria, and that means the genetic information of the virus can be moved to the E. coli genetic information. And that's how viruses actually replicate, by introducing their own genetic information into a bacteria or into any other living cell, including our body. So they use E. coli bacteria infected with a virus that consisted largely of protein heads and or coated surrounding DNA. So nothing basically major. Now, They also grew a batch of viruses in a medium, which is medium basically means their food, that contains two things. One is S35, and the second one is P32. What are these? So first of all, anytime we talk about these things, we're talking about radioactivities, which is really important, guys. Radioactivity makes it very easy for us to visualize the results of these experiments. So when we have 35S, the S stands for sulfur. Sulfur is help us track proteins because none of the DNA, none of the nucleotides that we have in our body contain sulfur. So if we have anything marked with sulfur, it means that's identified as protein. On the other hand, the P stands for phosphorus. Phosphorus is one of the essential molecules that are found in DNA. It's the backbone of our DNA, as I mentioned earlier. However, proteins do not contain phosphorus, which means anytime we have markers, which is 35S, that would mean that that's the presence of proteins. And anytime we see 32P, that means the marker for DNA. So what they did is they did a blender experiment that shows that viruses are actually transferring their DNA and not the protein components, which means, again, we're looking at more of the 32P residues being passed on versus the 35S, which is the proteins. Now, this is important because, again, it's encoding that the genetic information is the one that gets passed down from the parents to the offspring, not the proteins. Here is what they actually did in the experiment again to kind of help you understand it a little bit better. So in this picture on this side, you have the protein coded with the radioactive markers. And on this side, you have the viral DNA label, which is we're using the phosphorus 32. So notice that when they take the virus and infect the bacteria, so the little orange things are your bacteria, sorry, and these little ones are your viruses. So if the virus infects the bacteria, you blend it, which basically means you're breaking the cells open to get access to the viruses that are inside. You notice that they're completely separated. So you have the viral protein code that means the virus does not have does have the protein code, but the bacteria does not have it, which means that encoding, that genetic encoding was not passed to the E. coli, right? So the protein was not passed on to the E. coli. But in this experiment, when the virus attached itself to the E. coli and injected its genetic information into the E. coli, and when you break these cells, when you break the E. coli and look at inside of what it is, you notice that the radioactive marker is at the bottom, which is where the E. coli's are, which means that genetic information that was in the virus, right? The genetic information that was in the virus was actually passed down to the E. coli. Versus in this one, the genetic information that was in the virus was in the virus, but was not passed down to the E. coli. So this is extremely important for our understanding because it highlights that the protein component was not transformed or moved from the virus to the E. coli, versus the genetic information, the DNA component that was radioactively labeled, was transformed into our E. coli, which means that the hereditary pattern of the material or molecules is limited to the DNA and not the proteins. Now, there was, after they have this understanding of, okay, we do have molecules, these molecules do get passed on from one generation to another. We have an understanding, a basic understanding that they contain phosphate and they contain nitrogen and they have an acidic component. After we understood this, then there's some focus that was shifted to, okay, what is the structure of DNA looks like? So there was a lot of experiments done on that. So these experiments were basically reveal the structure and therefore the function of the DNA. And the importance of this, why do we care about this? First of all, this is really cool because first of all, it came from many number of disciplines. So it wasn't just, let's say, the biologists that were working on it. It's actually come from a lot of physicists, chemists work on it. This makes it very interesting because all the field basically contributed to what we understand from our DNA structure now. This is very fascinating because at this point, the molecular genetic component or a study of genetics didn't exist, right? So all the other fields basically contribute to our understanding of what the DNA structure looks like. So what is the DNA structure actually looks like? So one of the scientists that actually worked on it was the biochemist, a Russian-American biochemist, named Levine. And what he identified is that there is a 5-carbon sugar that is present inside the DNA. Now, this sugar, there's 2 types of it. One is identified as ribose, which is shown here. So you have 1 carbon, 2 carbon, 3 carbon, 4 and 5 carbon. Same with this one. So you have your 5 carbon sugar, but one of these carbons has an OH group. So we call that one of the sugars has a OH group. So we call that the ribose group or ribose sugar. But the other one is missing its O, right? There is no O here. So we're going to use the word deoxyribose. D as in lack of, you missing, and oxy as an oxygen. So basically, the difference between these 2 sugar molecules is that one is lacking 1 oxygen, and that's about it. So RNA, the R in the RNA stands for sugar ribose, and the D in DNA stands for deoxyribose. Now, the next thing that they understood is they also discovered that the 3 major parts of the nucleotides. And this is basically sugar, phosphate, and a base. Now, again, if you don't recall from our previous lectures, when we talk about nucleic acid, and I'm going to kind of hint at that over again, is the macromolecule. So this is our macromolecule. And the subunits, what this large molecule is made up of is small subunits called nucleotides. And what you see here, if I was kind of to highlight, this is one nucleotide. This is another nucleotide. Again, here's another nucleotide, another. And if I'm talking about the whole thing, then we call this the nucleic acid. So be careful with your vocabulary, guys. Now, again, they found that they have equal portions of sugar, phosphate, and base as they run the experiment. So first of all, they figure out that there's 3 components, and these components are in equal amounts. So what they deduce is that the nucleic acid has building blocks that must contain one of each of these components. Basically, 1 phosphate, 1 sugar, and 1 base. And I can see that by the color coding. Regardless of which sequence we're looking at, they're always going to contain a 1-to-1-to-1 ratio. Another experiment that was run by Charkov in 1955, which is the biochemist. Again, look at the chemist card, right? They're not all biology, they're also studying chemistry. They actually assess the ratio between the 4 types of nucleotides that we have. Now, again, the nucleotides labels are here, and I'm going to show you the list of the nucleotides later on as we talk about them. But we do actually have 4 major type of nucleotides inside our nucleic acid, and they are adenine, which are identified with letter A, thymine, which letter T, guanine with letter G, and cytosine with letter C. And when they actually assess the ratio of these, they noticed that the ratio of A to T is equal to, then the ratio between A is the same as the T or very close. And then the numbers that you have for the guanine is very close to the number of cytosine. And what that means, it means that basically the A or adenine will be base pairing or matching with a T, and a C will be matching with a G. To kind of looking at the number in humans, here are the numbers for the ratio of A to T, and then here is for the C to G. So this is important because, again, we're observing the relationship that A and T are base pairing together and C and G are base pairing together. Now, going a step further, we have in 1952, Rosalind Franklin and Wilkins were working on identifying the structure of DNA and figuring out what the DNA structure actually looks like. And they actually use a technique called X-ray diffraction. Now, we're not going to go into understanding what this process looks like, but basically what they understood is that they, first of all, let me show you the picture. Here's the picture that they took, part A, which doesn't seem a lot, like a lot, but based on the image that they developed, they say that the DNA structure is an actual double helix. And they have symmetrical organized subunits, which means basically they have a backbone, and then the rest of the structure form a double helix, which is the typical drawing that we have forming a double helix. And again, I'm not that good at drawing this, but here's the idea behind it. So Rosalind Franklin and Wilkins were able to do that by doing hundreds of hours to develop what they call photo 51 of DNA. Unfortunately, Rosalind Franklin never got credit for her work and contribution to identifying the 3D structure of the DNA. The credit went to two biologists and physicists in 1953 named James Watson and Francis Crick. You can see them by them looking at their 3D image they create based on the information that was collected by Rosalind Franklin. And basically what they said is that the 3D structure is a double helix, and they created what you see right here in the middle, which is what we actually study for the DNA structure. So they did a pretty good job deciphering what that DNA structure actually looks like. So this is basically an overview of what we already talked about in terms of who and what scientists contribute to our understanding of DNA today. Now, because we are again focusing on DNA, we're going to go a little bit further and talk about more specifics of the DNA structure. And some of these may seem repeated, but our goal is to make sure that we are clear before we kind of dive into more details of information. So the DNA structure. So the DNA structure basically, again, is the genetic material that can form multiple structures at different levels. The one that we are concerned about are basically genes, which is little segmentation of the whole DNA, which encodes or expresses specific proteins. The genes can be condensed or supercoiled to form our chromosomes. And all of the genetic information together will form our genome. So if you're looking at the gene, again, kind of going back to this, I'm always going to ask this question, and I want you guys to make sure you're very comfortable with this. What is the purpose of having genes? Again, genes are these little segments of our DNA or sections of our DNA that encodes for a very specific sequence of amino acid and lead to a synthesis of a very particular protein. We already mentioned this, but again, kind of highlighting the DNA is made up of nucleotides, which are the individual units. We have within a nucleotide, we have a deoxyribose sugar, if we're talking about DNA, and then again, ribose sugar, if we're talking about RNA. We have a phosphate group, and these are actually considered the backbone. They do not change at all. It's consistent for both RNA and DNA, and regardless of the base. And then for base, we have two types of structures, and then four general nitrogenous base. So we have These structures called purine and pyrimidines, and I'll show you the picture of these. So purines, which is these two hexagonal and heptagon that combine together to form our base. And this is guanine and adenine. And then our pyrimidine is a hexagonal unit, which makes our thymine and cytosine. I do not want you guys to know what this structure looks like. I don't even know how to draw them, but I do want you guys to understand that they do have different building blocks when it comes to the bases. So the bases are not identical to each other also. When we're looking at our chain of nucleotides, which is again forming that double helix, we're looking at a sugar phosphate backbone. Here is our phosphate, if you want to kind of highlight it. Here is our ribose sugar, again, a 5-carbon sugar. They form the backbone. And then what you see in blue color would be your bases. Now, because the DNA is a double-stranded structure, it's forming and aligning together to form a double helix. Based on the previous experiment that is done, we already know that the A is base pairing with T and C is base pairing with G. One thing that I do want to mention is that the little dashed lines that you see right here are actually called hydrogen bonds. And hydrogen bonds are basically the structures that hold the double helix together. So if there is, again, any damage to this double helix hydrogen bonds, then the double helix does fall apart and degrade. Now, the number of bonds present between the A and T is 2 hydrogen bonds, and the number of bonds present between C and G are 3. So in a sense, it's easier to break down regions that have A and T together versus areas that are rich in C and G connection. The other thing that I want to mention, if you look at the picture, is you notice the concept of anti-parallel sequences for the double helix, which means one sequence starts with a phosphate that is connected to the 5th carbon. So the 5th carbon would be right here, and ends with the phosphate that's connected at the 3N, which is right here. So this is the 3N. And the opposite strand, it starts with the 5 prime, or the phosphate connected to the 5th carbon, and then ends on this one at the 3 prime. So that's what they mean, anti-parallel sequence, where basically one goes from 5 prime to 3 prime, and the other one goes from 5 prime to 3 prime backward. Here is basically what we're talking about. Again, phosphate connecting to the 5th carbon makes it the 5 prime, and the next phosphate will be connecting to the 3 prime at the bottom for the first sequence. So here is the first sequence, 5 prime to 3 prime, and the second sequence would be 5 prime to 3 prime. Okay, so and I think this is well, the pictures basically shows you the numbering of the carbons. So 1, 2, 3, 4, and 5. Now, I don't expect you guys to go into a lot of details in terms of the structure. This is not a molecular biology class. We're just looking at a basic understanding of the genetic components. So again, DNA is directional, which means one strand, as I mentioned, goes through 5 to 3, as you can see here, and the opposite strands goes to 5 to 3. And I highlight this again, the dashed lines are your hydrogen bonds, 3 for CG connection and 2 hydrogen bonds between the A and T. The complementary base pair is another thing that I talked about. It's a complementary base pair between pyrimidine and purines. So A and T are pairing up together, and C and G pairing up together. So here is the depiction. Again, number of the hydrogen bonds that hold them together. So C and G are harder to break apart versus A and T. Now, this is not a new concept. We have discussed this in detail when we're talking about the condensation of DNA in terms of cell structure, or even the first lecture. So we understood that the double helix of the DNA can be coiled and wrapped around this protein called histones. And histones are these little circular proteins. And they form basically this bead on a string like a structure, which is what we have here. So here's our double helix. Here is those little circular protein that are histones. And the combination of each of these histones with its DNA wrapped around it is called nucleosome. So if we even take this a little bit further and keep condensing the genetic information, keep coiling the DNA, now we form a structures called chromatin. Now, chromatin is still small enough that you cannot, so it's about 30 nanometer. It is still small enough that you cannot visualize it under a typical light microscope. But notice that we keep coiling. So as we go farther this way, we keep coiling our structure to make it more condensed. So as chromosome coils farther and farther, now you are developing something called a chromosome, which is a typical X shape that you described. Now, I do want you guys to recall that when we do the X on the chromosome, they're actually made up of two sister chromatids, which is the identical copy of one another. So again, here, if I was to highlight, this is one chromatid, this is the second chromatid, and each of these are basically identical genetic information after the genetic information has been replicated. So this is a replicated genetic information in an X format, basically. Here's another depiction. Again, we have the double helix by itself, super fragile. We want to protect it. You wrap it around the histones and you form your nucleosome. Condense it even further, you get your chromatin fiber. And after many coiling, you finally get your chromosome, which has that X shape to it. Now, DNA, again, as I mentioned earlier, is super condensed, right? This is, again, another point that we're going to keep hearing about it. So when chromatin is looping itself, it actually, it's not condensed to the chromosome at that point. It formed these little loops by itself. That's about $10,000, sorry, 10,000 places in the genome. So they are pretty large sections of genetic information. So CTCF, which is an anchor protein, you don't need to know what that stands for. It brings in these sections together and create these loops that you see. So right here, this little purple structure is your CTCF protein. And again, by bringing two pieces of DNA together, you create these loops right here. So this is one loop. Let's say this is another loop. Now, the areas that does have these loops, they're called loop domain. So it means basically they are forming loop. And the areas that are missing that CTCF protein, they're called non-loop domain. So you do have both of them present within a condensed version of the DNA. So we learned about DNA, we learned about its structure, we learned that DNA is double helix. It's made up of nucleotides. The options for the nucleotides are A, T, C, G, which is basically either a purine or a pyrimidine structure. We understood that a nucleotide has a sugar phosphate backbone. The sugar is missing an oxygen, therefore is deoxyribose. You have connections between the double helix, which is the hydrogen bond, and the hydrogen bond, there's 3 between C and G and 2 between A and T. We also learned that for the genetic information is the only components that is inherited. Proteins do not, which was a concept prior to the experimentation. We have scientists such as Rosalind Franklin and Watson and Crick combining their information to create the 3D structure of the DNA as we know it today. And basically understand that DNA does get condensed first to form nucleosomes, then chromatin, and then finally the most condensed version, which is the chromosome. Now, one of the characteristics of genetic information is also the fact that it can replicate itself. So not only gets passed down from generation to generation for the purpose of gene expression or protein synthesis, but it also has to have the ability to replicate itself, which is the portion that we're going to basically focus on for today. So it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for genetic material. So as Watson and Crick are developing their own mechanism and understanding of the 3D structure of the DNA, this is one of the statements that they bring up, that basically the DNA that you see, it has the capacity to replicate itself. Now, one of the first things that you need to understand about the DNA is the concept of semi-conservative nature. So if you look at the picture that I have up here, this is the parent copy, and you notice that both of these are blue. Now, the concept of DNA replication, which basically means that you're doubling your genetic information. Some people may think that the parent would remain untouched, and then you basically create the second set of genetic information. But the concept of semi-conservative implies that the two new daughter genetic information that's created, they always keep the original copy, one copy of the parent's genetic information, and the second one is the one that is created through the process of replication. So semi-conservative basically means it's half conserved, which means comes from the original DNA sequence, and the other half was created through the process of replication. So in 1957, a couple of American geneticists, demonstrated the concept of semi-conservative nature of the DNA. And I have linked a video for you guys if you want to take a moment to watch it. It's a really cool video that kind of highlight the experiment I'm going to describe to you right now. So they actually they labeled the nitrogen base that are present in their experiment, they use what we call a heavy form of nitrogen. And they were able to trace how these nitrogen basically were utilized during the DNA replication. Now, higher density nitrogen, which is the heavy nitrogen, was incorporated into one strand of each daughter cell after the double helix was created. So by tracking the heavy form or the higher density nitrogen, they noticed that strand, half of it gets carried on forward to the new DNA that was created after replication. So here is basically what they mean. So here's the original sequence. And then when you do see the semi-conservative, you notice that the original was not that heavy, right? And then as you combine it with sorry, it was the heavy one, but as you combine it with the regular genetic material, it became lighter in weight or density, because now it's half of the original weight because it's semi-conservative. And as time passes by, you have less and less of that N15, which is the original sequence, and more of the nitrogen that was free floating in the sequence. So as more and more sequences were created, less and less of the parent sequence was present by the N15 marker and more of the N14, which means that the original sequence was remaining, but as the new genetic information was produced semi-conservatively, we did not change the amount of the parent sequence, and we maintain it at 25% after 3 generations. So the idea behind this experiment, again, is for us to understand that semi-conservative nature of DNA is extremely important. That every time the genetic information undergo replication, 1, 2, and 3, you keep one strand as the parent strand, and you use the parent strand to create the second strand of the double helix from the material available to the cell. Now, when we're talking about DNA replication, I apologize, DNA replication, why do we even care about DNA replication? So DNA replication is required when a cell undergoing cell division, which basically means if I'm taking one cell and making either two cells or four cells, depending on the type of cells, you need to double your genetic information. So this doubling of the genetic information takes place in what we call an S phase of interphase. So interphase is the big phase, and the subset of that is the S phase. Now, if you recall, in humans, we have 46 chromosomes. And as the replication happened, that number goes up to 92 during the process of cell DNA replication. I'm not going to talk about the rest of this cell cycle. We have a full lecture on mitosis and cell division of the somatic cells, where it goes into details about all of that information. So DNA replication. When our DNA is being replicated, the double strand first needs to be pulled apart. Because if it's in a double helix format, if it's in, sorry, oh, I lost my, if it's in a double helix format, then you won't be able to do anything with it, right? So it needs to be open. I had to, don't disturb my device. I apologize for that. So when DNA is replicated, we first need to open up our double-stranded healing, which is basically, as you can see, it says DNA is unzipped. So here is the part that it still needs to replicate, and this is the part that we are replicating currently. Now, if you notice, the DNA will always replicate itself from the from 5 to 3. So if you have a strand that starts with 3, right, the 3 prime, the new strand that's created is going to be 5 prime to 3 prime, which is what we expect to have. But the strand that ends with 5, it basically needs to start from this side, right? So it needs to start from this end. But notice that this part of the genetic information is still in a coil format, right? It hasn't opened. So what the DNA does is, as this DNA is unwrapping, I form these little fragments. So I form one right here, and then wait for the DNA to open up a little bit more. Then I form another fragment. Then I form for DNA to open up a little more, and then I form another fragment, and so on and so forth. So we actually create 2 strands, one that is continuous or consider the leading strand or copying from the leading strand. And the second one, which forms these little fragments of the DNA, is coming from what we call lagging strand and form its newest strand 5 to 3 prime at discontinuous pieces, or little Okazaki fragments, as they call it. So here is again the depiction of the concept of the genetic information being unwrapped, and describing this again. You have the parent cell, the parental strands are unwinding, and each parent strand is going to be used as a template to form the new strand in a form of semi-conservative nature. And always remember, A is going to base pair with T, and G is going to base pair with C. And the phosphate and sugar backbone is going to remain the same. I'm going to pause the lecture for just a minute, and I'll be back. My apologies for that, guys. So we're going to go ahead and continue our lecture. Okay, so when we're talking about DNA replication, we do need to have a group of enzymes. So there is this picture, and I really like it because it helps you visualize the enzymes that are extremely important for effective process of replication. So the first one we have is helicase. So again, look at the name. Helicase implies the helix. So it's a protein, it's an enzyme that basically unwind the beautiful double helix structure that we have. We have binding proteins that are depicted right here at this little purple blob that basically make sure that this double strand that I just popped open actually stays stabilized. Because remember, DNA can be very fragile, and if there's damage to this, you basically start to degrade your DNA, and you never want that to happen. So I have binding protein that is stabilized the separated strands. You have primases. Primases are basically responsible for adding these little short primers, which is the starting sequence of a nucleotide to the template strand. And then DNA polymerase, which is sorry, it's missing the word polymerase. So polymerase is adding the newest strand in a form of semi-conservative. So basically adding individual nucleotides to the parent strand to make the new strands. And then ligase. Ligase is like a little glue that takes those little Okazaki fragments that I mentioned and connect them together and make sure that they are sealed, and you have a beautiful, well-connected phosphate sugar backbone for the newly created DNA sequence. So let's look at this process in a step-by-step process, step-by-step for what goes on to replicate your genetic information. So first, as I described, your helicase is going to, which is this protein right here, is start to unwind the double helix for you. Then you want to make sure that the proteins are secured in their place. Sorry, not the protein, the DNA sequence are secure. So you basically use the binding proteins to stabilize your genetic information. And then you have your primases, which basically start or connect what we call a short strand to initiate the process of replication. And those would be, I'm going to draw exclamation mark next to them. So those would be basically, again, initiating the template for starting the genetic replication and allowing a point of attachment for your polymerase, your DNA polymerase to connect to. So DNA polymerase eventually comes in, which is, I'm going to show it. I can think of a shape, which is really bad, actually. So this is my DNA polymerase. I'm going to just kind of highlight it for you guys. So DNA polymerase comes in, attached to that starting sequence, which is in green, and it starts to create its sequence by adding new nucleotides and creating the semi-conservative strands of DNA. Again, I want to highlight this. This is my continuous, and this side would be my little discontinuous sequence or Okazaki fragments. DNA polymerase also proofreads the activity and make sure that whatever bases that are being placed is correct, and then make sure that if there is any incorrect bases, to replace them. Last, the continuous strand that I mentioned already, which is the top strand for us right here, is going to be synthesized from 5 to 3 direction. The bottom strand, which is right here, is going to be creating the Okazaki fragments or discontinuous sequence to make sure that the 5 to 3 prime pattern of replication is followed. After we're done, the top sequence is ready to go. The bottom sequence, however, needs to have the little pieces glued together using your ligase enzyme, which is right here. And this ligase enzyme basically, again, connects the sugar phosphate backbone of these Okazaki fragments together. As you can see right here, there's a connection point, and that allows the DNA sequence of the bottom strand to also be one big continuous helix. The cool fact about DNA replication is, if you want to think about it, is it doesn't happen at one end and then finish at the other end. So you actually create what we call replication bubbles, which you can see right here. So there is 3 replication bubbles right here. And what these replication bubbles do is that they each start their own portion of the DNA replication. And as these bubbles basically grow, the new double strands that are being created eventually connect together. So here's basically growing of those bubbles. And here's when that final double helix has been completely replicated, and you're ending up with that, again, that semi-conservative nature with two identical DNA helixes at the end of the process. So I think the first time I learned about DNA replication, in my head, I was thinking of kind of like this really long train ride, very long time for it to happen, because you can basically, there's no shortcut. But in reality, the shortcut is by you creating these little fragments of replication using these replication bubbles and expanding it and then eventually connecting it together. So think about it in terms of railroad. You have each city working on their own railroad, and then eventually, as they expand that railroad system, they merge together and form a very long, beautiful, continuous railroad track. Now, we understood the replication. We understand that you start with a double strand. The double strands open up. You secure it. You add the template. You add your DNA polymerase, and you start to create your semi-conservative double-stranded DNA. And when it is done, you end up with genetic information, or two daughter DNA sequence, which is going to be used during the process of cell division. But the ability of cell undergoing DNA replication is not limited to the cell itself. So we can actually complete the process of DNA replication or amplification in tiny test tubes. And that's basically referred as polymerase chain reaction. For this reaction, we basically provide anything that's necessary for the process of replication to happen, which includes the polymerase, the DNA polymerase. We add the nucleotides A, T, C, and G, and we basically create this machine that undergoes the heating and cooling system, a mechanism that basically unravel the double-stranded or the double helix, add the new nucleotides, and then repeat this process over and over and over again. So you are basically using this PCR mechanism. You can yield more than 10 billion copies of the DNA that you start by doing like 30 cycles of PCR. PCR is a tiny machine, is smaller than your typical, or it's about the size of a typical laptop. You use very small amount of liquids in micrometers to add to it. Again, you add the components, you set up the cycle as many times as you want. So typically about 30 cycles. And then at the end, you have created about, again, 10 billion copies of that target DNA sequence. This is extremely important if you're doing any type of forensic investigation, that you have very limited or very small sample to work with. So PCR allows you to replicate that genetic information and basically work with something that's much larger in the sample size versus a very small amount. Here is the basic idea. Again, this is happening in a machine. You have your DNA sequence, the one that you want to replicate. You add your polymerase, your DNA polymerase. You add your free nucleotides, which are again A, T, C, and G, and then the primers that basically initiate this. These primers are actually created specifically for your DNA sequence, by the way. So these are manufactured. And then all you do is basically, again, doing a temperature shift. You increase the heat and then decrease the heat. And during that process, you unravel the double helix, you add the nucleotides and the primers, and then everything goes back together. So original strands become 2, and then 4, and then 8, and then you basically multiply this by 2 every time you go through the cycle. Now, again, the steps of this process, we already kind of talked about this. I'm not going to repeat this. So preparation, you need the nucleotides and the primers, as well as the polymerase, the temperature shift going up and down, hybridization, which is basically means that the complementary primers and nucleotides are attached to it. DNA fills in the gap with the nucleotides, and then you repeat the process. You amplify it over and over again. But again, I already kind of mentioned that the PCR is really important in forensics analysis and basically looking at sample sizes that are very small. But what else can we use this PCR for? PCR can be used to amplify genetic information from animals that have gone extinct. So if you have very small samples in fossils and etc., you may replicate and amplify it. Microorganisms that cannot be openly grown in a laboratory, you can amplify the genetic DNA from them. You actually have cells and DNA from a 7,000-year-old human mummy. You can look at those cells and look at the genetic sequence of a 7,000-year-old human. You can look at the DNA sequences and of basically animals and see how the basically food consumption has affected genetics. They are very important for identifying any type of cadaver, yet it be roadkills, humans, bodies that have been damaged significantly. You can genetically alter bacteria that are basically going to be released for a specific test, and then look for the dispersion patterns of that genetic information throughout the space. You can, I don't know if that's a necessary thing, but you can eat poached moose meat in a hamburger by expanding the DNA and again utilizing that to make that hamburger meat. And then we can also use the genetic information for identify a specific human being and their connection to other relatives. The DNA sequencing that comes up usually after the PCR process is also very important. So if you have amplified the genetic information in, for instance, a rape kit, and you want to see who they're actually related to or who actually caused the, who's the culprit in that process, we need to actually sequence the DNA. So basically that means that sequencing individual genes and then check for the accuracy of the selected sequences within the genome. We can deduce a DNA sequence by aligning pieces of the different pieces of the genetic information and basically finding a match. So think about it as a puzzle. You finding one piece and you kind of matching it to a specific region of the genome. This is important. I think it's sequence of DNA is very important for us because it allows us to first again utilize that in terms of forensic investigation by matching the sequences of DNA that's been gathered from somebody who have suffered from any type of event that has left DNA behind. You can use it to basically take a DNA sequence, take a genome, and take little pieces of that DNA and try to overlap it millions of pieces together by finding these little pieces that fit in together. So let's say the first sequence, and I'm going to attempt to draw this. Oh, here, actually, I have a picture of this. So if this is a sequence, let's say the first line is tell us it's a T. There's another line that's a little bit higher. It's a T and A. And then another one, because it's getting longer, it's now T, A, and G. And then again, another line within our sequences sequencing of our DNA is TAGA. And again, as you go farther and farther, you're connecting these fragments together and eventually figure out that the full sequence is that. This is something that happens a lot faster today compared to what it used to be because of the technology in computers. But the idea here is if I have this piece, right? And then I have another piece, and then I have another piece, and I have another piece, right? So you start with this piece, and then these two pieces overlapping, right? So I know one piece, and then this piece, and then these two pieces are overlapping. So I know 2 comes, and then 3 comes after this. This overlap tells me that this section is next, right? So the overlapping region help us to figure out how I can sequence these little pieces of DNA to form the complete sequence of the genetic information. This is what the data would look like if you were to assess it. Notice that there are 4 different colors. Each of the colors are indicating a specific nucleotide. So for instance, a T is identified by the color red, G is represented black, and A is for green. And again, anytime you have that kind of peak forming, it implies that a specific nucleotide is present in that area. Now, it does take a lot of effort for the scientists to read this, but again, luckily, all of this is now done by computers, and all you do is basically set a sequence of DNA to companies, and the companies do this and then give you a sequence. So you don't have to do anything manual anymore, which makes it a lot easier. Okay, so that's about it for our today's lecture. So if you guys do have any questions about the content, please send me an email. I'll be happy to help you guys out. And otherwise, listen to the lecture, complete your Canvas post 2, and then just a note that your exam will be coming up at the end of this week. So please make sure you're reading out your announcement. Thank you guys, and have a good one.